Thank you very much for the introduction. Do you see my presentation mode, or are there the slides in total?
Yes, we do.
Yeah.
Yeah.
Okay, good.
Wonderful.
Thank you. Just putting this away. Sorry. Yeah, so very happy to be here to talk about what we're doing at the university and at Faron Pharmaceuticals. So as mentioned, I'm the Chief Scientific Officer at Faron, but I also have an academic lab at the University of Turku. And we are under this InFLAMES Flagship that is funded by the Research Council of Finland, where we do all the related investigations on immunity and how to utilize immune cells for therapeutic benefit. So I will be talking to you about our research on reprogramming macrophages for therapeutic benefit. And this is work done for many years in my lab and with collaborators. I'm happy that Andreas already told you a lot about macrophages, so I won't be going into detail on tumor-associated macrophages, but they are very versatile cells, and they do many things to promote tumor progression and metastasis. So the way I think about macrophages and how we could reprogram them in tumors to activate innate and adaptive immune responses comes to their functions on scavenger receptors. And so the scavenger receptors are scavenging different components in the tumor microenvironment that then promote tolerance in macrophages then to support tumor growth. There are also many different other components that by agonistic antibodies, you can stimulate their functions to activate anti-tumor effector functions. But I will be talking mainly about the scavenger receptors because we have been working on those. But the main point is that by either inhibiting scavenger receptors or other receptors affecting the tolerogenic macrophage phenotype, or by activating the anti-tumor effector target molecules, we could be possible to increase antigen presentation on tumor-associated macrophages and thereafter recruit CD8 T cells and NK cells and induce killing and phagocytosis of cancer cells via macrophage eating. This is a review article, systematic review that we put together with my PhD student in 2024, because we wanted to understand the clinical landscape of macrophage reprogramming therapies. So what is done recently and what are the different target molecules? So we screened over 700 trials with a bit less than 200 different drug target molecules. And you can see from here that this research and clinical trials has expanded significantly from 2010. And the most contributing targets in this field is the CD47 SERP alpha axis. There you can see a lot of clinical trials, but you have also toll-like receptor agonists, also the LIL-RB layer pathway, and also IDO1. There hasn't actually been many very successful trials. Macrophage reprogramming therapies don't normally work very well as monotherapy, so you need the T cells there to be activated. And also there are very many complementary pathways that if you inhibit one, you activate the other one. So therefore, it's not very efficient to only block IDO1, for example. And it was interesting when we went through these studies that there were 6,000 patients treated with an IDO1 inhibitor before they discontinued the development. So it's really massive effort that they have put into this. And I want to also highlight that many people are investigating the same target. So there are many companies around CD47 SERP alpha inhibition. But also these therapies have many side effects. And this is the main cause why some of the trials have been discontinued. We have been working with Clever-1, which is a scavenger receptor expressed on macrophages and endothelial cells. So the name comes from common lymphatic endothelial and vascular endothelial receptor, but it's also known as stabilin-1 and FIL-1, and I will tell you why it has three names in a while. But on macrophages, Clever-1 expression regulates macrophage tolerance. It resolves inflammation and causes immunosuppression in tissues. But it also regulates tissue homeostasis by scavenging bacteria, apoptotic cells, and endogenous substances. On the endothelial side, it regulates trafficking. And here I want to tell that in the initial research done in the University of Turku identified that Clever-1 is regulating also cancer cell metastasis to the lymph node. So the cancer cells were utilizing Clever-1 as an adhesion molecule to metastasize and colonize to the lymph node. And also it regulates scavenging of endogenous substances of the liver sinusoidal endothelial cells, but also extravasation, rolling, and binding of leukocytes. So based on the initial findings that Clever is expressed on endothelial cells and macrophages, and that it was regulating tumor growth. So by blocking Clever-1, we could see that the tumor growth was diminished and metastasis as well. When I was starting with this molecule and starting my lab, I wanted to actually understand what was the main component driving these Clever-1 actions. So we utilized Clever full knockout mice, or then these knockout mice that were where we had Clever-1 knockout specifically from macrophage population, so the LUS2 Cree model. And when we implanted Lewis lung carcinoma cells or triple negative breast cancer cells, we could see that the ones that were where Clever was absent only from the myeloid cells didn't grow tumors at all. We went in more detail to look what was happening in the tumors, and we found out that it was filled with CD3 positive T cells. And when we did a more phenotypic search with flow cytometry, we could see that the infiltrating T cells were mainly CD8 cytotoxic T cells. And here I want to also emphasize that if you look at the middle graph here, or the Clever full knockout, it has some T cell infiltration, but in mouse, because Clever-1 was regulating the leukocyte migration within endothelial cells and lymphocyte migration, that it might be that due to the reason that Clever is absent also from the endothelial cells, that they cannot be fully infiltrating into the tumor, and that takes just more time. So now I want to go back to history a bit, and how did we end up on working with Clever-1? So initially, Sirpa Jalkanen at the University of Turku was investigating molecules and trying to discover how were lymphocytes going into the lymph nodes and exiting the lymph nodes. So what she did was that she took lymph node lysates from human preparations and isolated lymphatic endothelial from cells. And then what she did was she immunized mice with this lysate and then isolated B cells to do hybridomas. Then she was screening these hybridomas for antibodies that were specifically binding to lymph node lymphatic endothelial cells. So then they published this story about this one antibody that they identified here, which was called 3372, and it nicely stains the lymphatic endothelial cells in the lymph node. And Heikki Irjala at that time named the molecule Clever-1. But at the same time, there were two other groups, one from Japan and one from Germany, that they discovered this molecule on macrophages that was a novel scavenger receptor affecting bacteria scavenging that they called FIL1. And then Gerd from Freiburg, if I remember correctly, he also identified stabilin-1 and 2 that had very similar structures, and they were expressed on these immunosuppressive macrophage populations. So how did we go from this original finding to the immunotherapy drug that we now are developing at Faron Pharmaceuticals? So that was the first initial study at 2003, where they identified that Clever-1 regulates binding of cancer cells to the lymphatic endothelium. In 2007, Faron was built up by university spin-off. And what was done for the antibody that they discovered in the original paper, this 372 antibody, is that they humanized it and de-immunized it with antitope. So it was converted from a mouse IgG1 to a human IgG4. So now it's called Bexmarilimab, and then 2 point mutations were done to the Fc part of this antibody. So the first mutation was done for the reason that to prevent Fab arm exchange. So IgG4 antibodies are known to change Fab arms. So that was inhibited by the first mutation, and the second mutation was done to dampen the effector functions that this antibody has. So what is known about antibodies is that these IgG1, 2, and 3, depending on the type, they might have antibody-dependent cell cytotoxicity and complement activation. And the IgG4 has the least of those. And if you do those mutations still, then you can get totally rid of these effector functions. And the reason why we wanted to get rid of these was that because CLEVER1 is also expressed on endothelial cells, so that it doesn't cause any effector functions against those. So to reduce toxicity mainly. So then we went on and produced some academic, first of all, patents. So we patented the antibody. And then several publications came out from Siira Pajalkanen's group that was describing leukocyte trafficking and clever expression on placental macrophages. So there, CLEVER1 was really regulating the placental maternal and fetal interface, so that the pregnancy was going as it should. Then Annika Olgars was identifying clever expression and or high clever expression was poor prognosis in colorectal carcinoma. Then in 2013, the cell line was made that was producing Bexmarilimab, so Chinese hamster ovary cell line. And thereafter, this was the metastasis paper that I was briefly mentioning about, that clever can regulate metastasis of cancer cells. Then 2015, we did the primate toxicology studies and also the GMP production at Abtena. And also Senthil Palani in the lab, he did a discovery that clever can inhibit antigen-specific Th1 T cell activation. Thereafter, the first in-man trial was started, which was called MATINS. I will show you results of that, and that was in the end of 2018. And then this was the proof of concept paper that I showed you briefly, that the macrophages were regulating the clever phenotype in tumors. So how does this all work? So there has been a lot of work behind how can I draw this diagram for you? So these are the associated papers, but just to explain how Bexmarilimab works to inhibit clever function. So normally clever scavenges, let's say oxidized and modified LDL. And what this does for a macrophage, it promotes tolerogenic functions like lipid-associated or nuclear lipid receptors, and also it induces IL-10, promotes Tregs. And what this macrophage does when it's clever positive, everything it scavenges, it's kind of taking it into the trash. So it's kind of removing everything that would be immunogenic to the body, and therefore it can maintain homeostasis. And this is actually good for you in normal conditions, but you don't want these in the tumor. So now when we give macrophage Bexmarilimab, it binds CLEVER1 and it's rapidly taken into the macrophage. So first of all, the binding site where Bexmarilimab binds to clever is the binding site where acetylated LDL binds to. So it can prevent the binding of acetylated LDL or the scavenging of it. But it also, when it enters the endosomes, it impairs the endosomal acidification, which is depicted here. So it associates with VATPase, and when Bexmarilimab is present here, the VATP pump cannot work, so the lysosomal acidification doesn't occur. And this affects the processing and presentation of antigens on the macrophage. So this kind of represents how dendritic cells are like. So they kind of have a prolonged antigen processing due to this impairment in acidification, and this leads to improved expression of MHC and loading of MHC of antigens. What this also does is that it induces TNF-alpha, IL-12, CCL3, CCL4, CCL5, and CXCL10, and this has been shown in these publications. So this is now the results of the MATINS trial that I pointed out. And so what we found in this trial, which was a basket trial investigating 10 different tumor types. And I have to also mention that these tumors were advanced, so these were last line patients, so medicine didn't have anything to offer them anymore. And this was a single agent trial, so that Bexmarilimab was the only drug that were given to the patients. But basically, by Bexmarilimab binding to Clever-1, so the tumor-associated macrophages hypothetically can be activated and then induce these adaptive immune responses. So what we found out was that targeting Clever with Bexmarilimab was well tolerated, and we could achieve disease control in late-stage cancer with Bexmarilimab monotherapy. And you can see here depicted in red that the more the red the tumor type is, the better responses we saw. So in breast cancer, for example, we saw a 35 to 40% disease control. But what was also interesting was that if we look now of the biopsy samples, tumor-associated macrophages, so we utilized GeoMX, digital spatial profiling, to understand could we convert macrophages in the tumor to promote anti-tumor responses. So we saw nicely that in the patients who got a response, there was a TAM transcriptome activation. And this led to higher interferon gamma in the circulation during cycle 1 of the treatment and to prolonged survival in these patients. So this was a proof of concept that, yes, indeed, we can convert these macrophages within tumors to be activated. And also, surprisingly, what we found out was that low baseline immune activation was one that associated with the Bexmarilimab benefit. So the question now is also that, okay, macrophage conversion was happening. What happened to adaptive immune cells? So when we look at the circulation, we could see that there was a rough and robust induction of NK cells, CD8 T cells, CD4 T cells, but we saw reduced numbers of FOXP3 positive T cells. We also saw a nice induction of B cells in the circulation. Now, if we go to see what was happening phenotypically for the T cells, so this is mass cytometry of the circulating T cells. So here you can see CD4 T cell population and CD8 T cell population from 7 patients. And here you can see that if we compare now the pre-treatment to the post-treatment, so 7 days after giving first dose of Bexmarilimab, there is a massive activation of almost all the T cell subsets that we can find from the mass cytometry, as they were expressing activation markers for CD25, CXCR3, and CD69. And some of the negative checkpoint molecules, such as PD-L1, were downregulated, and also CTLA-4. So then the question is, because we saw that there were patients benefiting from Bexmarilimab, who are these patients? Because, of course, you need to define the patients who you want to treat to get a positive readout for a phase 3 trial. So first thing we looked at, of course, was immunohistochemistry for Clever-1. And we could actually see that the higher the intratumoral Clever expression was on macrophages, that was associating with the disease control in patients. What we also found out was that if the patients were low in PD-L1, they had a better benefit. So this goes along with the idea that you are suppressed. So the less amount you have interferon gamma at pre-treatment or TNF-alpha, the better response you will have by Bexmarilimab treatment. So therefore, we wanted to validate this kind of phenomenon that we were seeing in our patients. And as Andreas explained to you, that it's really difficult to get like representative cells for in vitro culture that represents tumor-associated macrophage heterogeneity. So therefore, we used ovarian ascites TAM. So we got ovarian ascites samples from the university hospital and actually treated them as a whole pool. So every cell that we got from it, we just put Bexmarilimab in that cell population and then looked what happened after 48 hours of exposure to Bexmarilimab. So here you can see what type of cells there were in the ovarian cancer ascites. So you can see here clearly that there are many, and there were cancer cells as well. And here you see the monocyte macrophage population here, which was also clever positive. The basic finding that we found for the responses was that there were patient ascites samples that were actually responding to Bexmarilimab the same way as they were responding to matins. So the Bexmarilimab sensitive ascites had very low levels of interferon gamma at pre-treatment. And if we added Bex ex vivo, we got an upregulation of CXCL10 positive TAMs and interferons up. Whereas the samples that didn't respond to Bex were those that had high interferon, high Treg, high population of IL-4 inducible 1 macrophages, and high level of NK cells proliferating. And actually, in this case, Bexmarilimab treatment took interferon response down. We concentrated our efforts to look at the IL-4 inducible 1 macrophage because that was an interesting population. What is known about IL-4 inducible 1 is that it's upregulated upon interferon stimulus, more so in chronic exposure of macrophages to interferons. So you can see here that in the ascites samples, the responding patients had lower levels of these. But when we went to look at matins biopsy samples and stain for IL-4 inducible 1 on macrophages, we could also see a trend that the responding patients had lower levels of these IL-4 inducible 1 macrophages. So to continue this, we wanted to kind of also understand, do we see, or in a more like treatment and tumor setting, we had the possibility to have mastectomy samples from the University of Turku and Turku Hospital, where we get tumor, fresh tissue tumor, and adjacent healthy tissues. And we wanted to understand, do we see similar responses after Bexmarilimab therapy, what we see in the patients and the ascites samples? And to understand that what was a response to Bexmarilimab, we utilized these gene signatures that we get from the matins trial, post versus pre-Bex therapy, and the ovarian ascites Bex versus IgG4 ex vivo treatment, and collected a Bex response signature that was found in both of these data sets in the responding patients. And so we treated these explants. Now there are roughly 20 patients, and we could see that some of the patients really get this nice response. So this gene signature upregulation that we found from both of these data sets. By doing area under curve analysis of these different gene expression changes, we could clearly see that there were 5 different genes that were highly upregulated in the responding patients, and this we call the Bex signature genes. And we can see with these 5 genes, we can clearly separate these 2 populations of responding and non-responding patient-derived explant cultures for Bexmarilimab treatment. Similarly, by measuring the cytokines that were related to Clever-1 blocking, we can see that there was only 1 patient sample that didn't go along the same line with the gene expression. So they were nicely correlating to each other. So we get a high upregulation of CCL4 and CCL5 in this setting. What was also interesting, and I wanted to understand that because in the monkey tox studies, we never saw any side effects or anything alarming. So we wanted to see how this Bexmarilimab treatment was affecting the adjacent healthy tissue. And it seemed to be that there was no consistent responding, non-responding in the adjacent tissue. So everything was behaving the same way. And here you can see the cytokine measurements, and you can see that in the responding tumor, that was the only place where we got this kind of inflammatory stimulus. And all the others, the adjacent from the responding patient samples or the adjacent non-responding, were pretty much looking the same as the tumor that didn't respond to the treatment. So we wanted to understand why this was. So we went to look with single-cell RNA sequencing, what kind of monocytes and macrophages were actually in the breast tumor tissues that we were treating. So here you can see that we can find from the adjacent tissue, you find a certain nice of cells of different flavors, kind of, if I can say so. And then there is the tumor tissue monocyte and macrophages, and you can see the clever expression on these cells here. So what was interesting to us is that in the adjacent tissue monocyte macrophage populations, most of them were actually tissue-resident macrophages because they were high in FOLR2, LY1, and MRC1. Whereas in the tumor, most of the cells were monocyte-derived macrophages having TREM2, SPP1, and CAD-M1. So what was changing then because what we saw was that CLEVER was actually equally expressed in resident macrophages and monocyte-derived macrophages in tumors. So what was the different factor then that was defining these responses between the adjacent and tumor tissue? And we could see that in the tumor tissue, there were TREM2 positive macrophages and IL-4 indicible 1 macrophages that were CLEVER positive. So that was the difference. But also we wanted to understand more the spatial context of this as well. So what we did, we went back to spatial transcriptomics and wanted to see where was CLEVER-1 macrophages in the adjacent tissue compared to tumor tissue in relation to other immune cells or cancer cells. And what we noticed was that in the tumor tissue, CLEVER-1 was mostly very close to CD8 T cells and many other cell types like B cells, CD4 cells, CD8 T cells, and so forth. Whereas in the adjacent tissue, CLEVER-1 was only adjacent to B cells, which is very interesting. So you can here see it like that. So what we concluded out of this experimentation was that you have actually 3 different responses that Bexmarilimab can cause, depending on what cell is close to macrophages or whether it's a low or high or a cold or hot tumor, depending on interferons. So here you can see that in the adjacent cancer-free tissue, the macrophage is close to a B cell, and what we saw in the publication in YITSI, that Bex can induce a low inflammatory response and B cell activation, and this is actually what we see in our patients. So there are some quite autoantibody formation, which might be coming from the healthy tissues that Bexmarilimab is affecting. Whereas in the tumor, if we have a late-stage activated macrophage, Bexmarilimab can actually downregulate the chronic inflammation. And normally this situation is the one that is responsive to immune checkpoint blockage therapies. Whereas if the macrophage is a monocyte-derived TREM2 positive macrophages, there Bexmarilimab can induce a moderate inflammatory response. And these are normally the ones that can then respond to Bexmarilimab treatment. What is also interesting was that because we didn't see any effect basically on the resident tissue macrophages in adjacent healthy tissues, we wanted to understand, can we actually reprogram also these macrophages so that we give them tumor conditioned media from the same patient's tumor and incubate that conditioned media on the adjacent tissue. So what we then found out that if we did this experiment, we could actually convert the adjacent tissue macrophages from the responding tumor media to a similar adaptive immune activation state that we saw in the tumor when they were treated with Bexmarilimab. So basically, this indicates that the tissue resident macrophage state in the adjacent tissue is not fixed. We can promote immune activation also via dose, but it still needs a component from the tumor microenvironment, being it a danger-associated molecular pattern or some other metabolites that then kind of increase the activation of our drug in this setting. But I want to just highlight now that we have learned also during the process of investigating CLEVER-1. So I told you about macrophage activation, and this was our like a cornerstone of how our drug works. But in the previous years, we actually discovered that macrophages can secrete, or endothelial cells can secrete CLEVER-1 in extracellular vesicles. And this exosomal CLEVER-1 can be a soluble checkpoint, where I will tell you a bit about that. And then we also found a couple of things, and I will go briefly also deeper to those as well. But let's go first to the very interesting study on the immunosuppressive activity of extracellular CLEVER-1. So during the studies, we screened the breast cancer, because we had a breast cancer cohort from which we were taking the mastectomy samples. And this is the MATINS patients, these solid tumors. And then we compared healthy patients. So we screened their plasma for soluble Clever-1 and found out that actually Clever is more highly expressed in the plasma of breast cancer patients. You can see in pink, and the MATINS trial patients in orange, compared to healthy donors, which is in blue. What we also noticed from the MATINS patient was that the soluble Clever-1, so we call it soluble Clever-1 or secreted Clever-1, that Bexmarilimab therapy could reduce significantly these levels. So you can see that it's disappearing from the plasma of, this is an example of a patient, but you can see here that the more you gave Bexmarilimab, the higher the decrease in soluble Clever-1 was in the plasma. So why is this important? So first of all, we were a bit puzzled, like why cancer patients have higher levels of soluble Clever-1. So what was inducing its secretion? So we went to see the cytokines compared, so cytokines in healthy donors compared to breast cancer patients, and identified several components that were differently expressed or in the plasma of these patients. And many of them were these inflammatory mediators or reflective of inflammatory conditions. So I kind of suggested that this is a chronic inflammatory state that then is releasing Clever-1 into circulation. What we also tested was, which is shown here, so we had our mastectomy patient samples, pre-operative and post-operative. So this is 3 weeks in between. And we wanted to test whether the tumor was secreting soluble Clever-1, but this wasn't the case. So it really didn't matter whether the sample was pre or post surgery. But what we noticed was that if a macrophage was stimulated with interferon or LPS, they secreted much higher levels of soluble Clever-1 than the immunosuppressive macrophage population. The similar thing was seen in liver sinusoidal endothelial cells, that if you gave them TNF-alpha interferon, they secreted much more of Clever-1. So what was this component that we were investigating? So this is electron microscopy images of immunogold labeling of Clever-1. And it's interesting that you can see Clever-1 vesicles outside the cells, some going here and here secreting like this. But you can also see this tunneling like nanotubes where you can see a lot of Clever. So Clever is actually communicating between two cells in this figure. So we did ultracentrifugation and found out that Clever-1 can be present in the plasma or cancer cell supernatant or lymph in 3 different formats. So you have soluble, totally soluble, you have exosomes, and you have microvesicles. So we wanted to know what is the species like that we can find for Clever. So we did Clever pull downs from the plasma. And you can see that it's heavily truncated in the C-terminus. So all the peptides that we can identify from the mass spec results was that they are heavily in the N-terminal side with 2 different antibodies that we used for Clever pull down from the plasma. And this was the dominant bands we can see, and they were mass spec analyzed. So what we did was that to kind of investigate this component better. This is Clever molecule, so it's very large. It's 280 kilodaltons in size. So we recombinantly made this first half of Clever that was representing or mimicking the size that we found from the plasma of endogenously present Clever-1. So we made this, which we call H1. And then what we found out was that Clever can bind, or this H1 can bind T cells when they get activated. So what happens when a T cell gets activated is that during the immunological synapse is that it flips its membrane, exposing phosphatidylserine. So you can see here that if we induce the intracellular calcium levels with ionomycin, you get an increased binding of annexin 5, which is binding to phosphatidylserine. But you also get increased binding of H1. And this is a transient binding. So when the immunological synapse then converts back, then you don't get binding anymore. So this is a very precise mechanism how Clever-1 can bind T cells. So why does this matter? So we first wanted to understand what molecule was Clever binding to. There has been some publications that Clever would bind phosphatidylserine, but we didn't see this. So when we incubated, pre-incubated with annexine and then incubated with H1, there was no blocking of H1 binding. So we did again a pull down of Clever-1 in Jurkat cells and identified actually that the H1 was binding insulin growth factor 2 receptor in the domains 1 to 10. Domains 11 and 13 are the ones that are normally binding insulin growth factor. So it was a different binding site for H1. So when we went to test our patient samples that can Clever or the H1 recombinant protein bind T cells, so we actually found this very nice population in the circulation PBMCs or T cells where Clever, the H1 was binding. So you can see in healthy donors, it was binding this population, and breast cancer patients, this population. And if we look at what was the cells that it was binding to, they were memory cells that were high in PD-1. So they were activated memory cells where this transient binding was occurring to. So what consequences this H1 binding to T cells has then? So if we incubate T cells during their activation with H1, we get an increase in FOXP3 positive T cells. So here you can see the Th1 transcription factor and the FOXP3 regulatory T cells. And this was via phosphorylation of LCK. So normally you get a nice activation of LCK when you just activate your cells. But in the presence of H1, this activation of phosphorylated LCK was diminished. And maybe this is causing the Treg upregulation. Also, we did this macrophage T cell co-culture assay to understand what kind of T cells we were now dealing with. Because, okay, we see FOXP3 positive cells, but what is the consequence if you now consider a macrophage cancer cell or a macrophage T cell crosstalk? So then we did so that we isolated, so this is autologous T cell macrophage assay. So we activated T cells with the anti-CD3, anti-CD28, and IL-2, and then incubated it with control or H1 for 6 days. At the same time, we differentiated monocytes with MCSF for 6 days, and after this, we then put them together to grow. And what this H1 incubated T cells did to the macrophages was that it decreased HLA-DR, CD40, and a little bit of CD163, but articulated heavily Clever-1. So it seems that it was a feedback forward loop that then was promoting immunosuppressive macrophages and Clever-1 on the macrophage. And finally, what we did was that, so what consequences H1 or the release of soluble Clever-1 in the tumor microenvironment would actually do. So what we did, we utilized our mastectomy samples and did this patient-derived explant culture so that we wanted to first see H1 binding on activated explants and what is the treatment response after H1 treatment. So first of all, we can see that the H1 was binding CD8 T cells only after they got activated. Also similar findings was with CD4, but what was very robust was that it was increasing significant amounts of TGF-beta in the tumor microenvironment. So basically the concept is that these Clever positive cells, macrophages or endothelial cells, are releasing soluble Clever-1, which can bind to this immunological synapse and prevent the T cells from Th1 activation and inducing FOXP3. And we also showed that it can induce or interfere with anti-PD-1 responses, so diminish that response as well. So, but in the last section of my talk, I would just briefly want to also show what also we have found. So I think an important factor is that Clever-1 is expressed on myeloid malignancies. So what we actually are now developing at Faron is the clinical development pathway in myelodysplastic syndrome, and I will show you why. So first of all, this is a hemap plot of different hematological malignancies, and you can see here that the more red, the higher Clever expression you have, And you can see high expression in AML and MDS, and these are the normal monocytes. But you can also see some expression in some lymphocyte leukemias. So what we first of all did, based on what we knew about this myeloid malignancies, we conducted a phase 1 to 2 trial where we had Bexmarilimab plus azacitidine for higher risk myelodysplastic syndrome or relapsed or refractory acute myeloid leukemia. And what we found out was that indeed, when we administer Bex with AZA, we can see an increase in HLA-DR on the monocyte population. So it was increasing antigen presentation, and this was reflective of the response that we saw in the patients. What we also noticed was that the CD3 positive T cells in the bone marrow were increasing in absolute numbers. So this was a really nice phenomenon that was kind of reflecting that what we were seeing in the solid tumor trial. What we also did to understand what was actually happening in the bone marrow, so we performed single-cell RNA sequencing on 5 responding patients. And here is how the UMAP plot looked like at entering the trial. So this is a screening sample. So you can see here monocytes to granulocyte myeloid progenitors and blast cells. And after 3 cycles of treatment, you can see there are clear, robust bone marrow remodeling after Bex and AZA treatment. So you can see that the immunosuppressive monocyte populations were disappearing, which had high TGF-beta levels. But we also see an increase in progenitor cells of erythrocytes, leukocytes, and platelets that were increasing in number. So based on what we knew about Clever-1 in these hematopoietic situations, we actually conducted a study where we utilized wild-type and knockout mice and gave them 1 dose of 5,4-Uracil. So 5,4-Uracil is a chemotherapy agent that can deplete your red blood cells and white blood cells almost completely. And then we can look at the recovery of our different cell types after this dosing. And what you can see is that the Clever knockout mice, even their bone marrow was much more red than the wild type animals. And you can see that there was a clear induction and robust recovery of white blood cells, lymphocytes, platelets, neutrophils in the knockout animals compared to wild type animals. We did the same thing with MSTAB antibody. This is a mouse surrogate antibody for Bexmarilimab. And when we dose prior to F5U and then dosing every 2 days, and then blood analysis at day 14, we can see that there is an increase in B cells, CD8 T cells, monocytes, and granulocytes after anti-Clever treatment. So it was clearly activating hematopoiesis in these mice. So going back to the trial, so the Bexmab was a phase 1 to 2 trial, and now we have completed the phase 2 part of the trial. And here you can see the overall response rate. So we saw more effects in the myelodysplastic syndrome, and therefore we went to investigate more on those patients. And you can see here the waterfall plot. And just to mention that there were patients who got complete response. So 45 patients got a complete response in treatment naive setting and 2 in the refractory setting. But there seems to be very robust effects on the bone marrow blasts, and also that we could bridge patients to transplant, which is the only curative treatment at the moment. And the median overall survival in refractory HR MDS or high-risk MDS was 13.4 months. Which is very good because normally it's around 6 months in this population. So this is very promising, these results, and we're continuing to go to phase 3 in this MDS setting. I also want to also highlight, this is something very interesting that we found out, that the better target engagement of Bexmarilimab with soluble Clever-1 in the bone marrow assays with complete response. So the better Bexmarilimab binds to soluble Clever-1, the better the patient gets a response. So I think the soluble Clever has a meaningful role also in these hematological malignancies in our patients, so that we can inhibit the presence of soluble Clever-1. And finally, I want to just mention that because the phase 2 was in combination with azacitidine, so how can we kind of dissect what was the effect of Bex and what was the effect with azacitidine in our patients? So we did RNA sequencing of azacitidine-treated MOLM-13 cells versus DMSO or Bexmarilimab versus isotype. And you can see here that they actually complement each other very well. So here, azacitidine actually can prime adaptive immune responses. You can see here, like leukocyte chemotaxis, leukocyte migration, adaptive immune responses go up, whereas Bexmarilimab can actually affect the blast cell mitochondrial ATP synthesis coupled electron transport. So it can actually prime the mitochondria for apoptosis. So they're synergizing together to induce these robust effects. So altogether, I would conclude that Bexmarilimab combined with AZA induces broad hematopoietic and immune reprogramming in the bone marrow. So it activates progenitor populations, enhances lineage commitment and niche normalization. It increases lymphocyte abundance and cytotoxic function and engages complementary epigenetic and metabolic pathways in blast cells. So all of our findings support the therapeutic potential of this combination in myeloid malignancies. So I will finish here and thank my whole lab and all the funding agencies, Faron, clinical trial sites and patients, also my collaborators at University of Birmingham and Turku University Hospital and University of Helsinki. So thank you very much for listening.
Well, wonderful talk. Thank you very much. An overwhelming long journey, I think, but also very positive, of course, and very successful journey. So the paper is open for discussion. So who has a question? I don't see the chat, but I'm looking for the, if a hand comes up, maybe I should otherwise start. I was wondering the Clever-1, the treatment, the Bex treatment. So what is the clinical schedule? Is it important that you give this on a regular basis? Because I think it's an interfering. So you also don't want to interfere with all the internal regulations within the immune system. So I can also imagine that at a certain schedule, you actually decompensate actually your effect. Did you look into that and what is happening there?
So in the solid tumor trials, we were giving the drug every 3 weeks. So basically the antibody is cleared very rapidly. So its half-life is around, depending on the dose, around 2 days or 3 days, but the pharmacodynamic effects last longer. But still within the frame of 3 weeks, I think we're going back to, the baseline. So in my opinion, we don't induce kind of a shift that it would exaggerate inflammatory responses to that extent that you start getting feedback mechanisms that then compensate for that. So I don't think this happens. I think we're just like boosting the immune system a bit so that it can activate the adaptive immune system, and then we're done basically. But I think like a boost drug dosing would be best. So not consistent or constant dosing, but kind of this boosting effect. Yeah. But if we consider now the hematological malignancies where the blast cells are also expressing Clever-1, there we have a weekly dosing, just to have the target engagement also on the malignant blast that also express Clever-1, that we can suppress their activity, and seems to be rather actually good. So I think you have to kind of think where you're treating and who you're treating and with what component you're treating, like combination therapies.
Yeah, I think so. And just to remind me, from the solid tumors, what combination did you actually look into? So what combination treatments have been done?
So that was only monotherapy. Okay. So only single agent, but we are starting investigator-initiated trial with chemotherapy and anti-PD-1 blockage.
Yeah. Okay, that combinations. Yeah. Thank you. So is there...
There was actually a very similar question in the chat from Alini again, because she wanted to ask you exactly if there's a perspective of setting up a clinical trial combining Bex with anti-PD-L1
Yes, yes.
immunotherapy.
Yeah, we have done mouse studies regarding the combination, and it seemed that it would have an additive effect in those tumors that are refractory to anti-PD-1 treatment. So we could then increase the response rate there.
And maybe I would also like to ask you now, seeing your amazing journey already and seeing what you've all discovered in the past just 10 years is really crazy. If you think then ahead, maybe in the next 10 years, where do you think this can go? Can this also, move even earlier into the treatment regime? So not just late stage advanced, but can we also dream of maybe a first line therapy?
We are hoping so. And actually in the naive setting, we were in first line, of course, in the hematological malignancies. That was first line treatment. But we are thinking of moving to earlier lines of therapy in breast cancer. We are starting a trial there. And so I think now we know that our drug is safe. So now we can move on to earlier lines, because I think one significant point that we need to understand with our drug is that we need the adaptive immune system to be there. Yeah. And if patients are like exhausting all treatments, they don't have working bone marrow, they cannot produce enough of T cells, we don't get any benefit from our drug. So we need the intact immune system to be there for tumor reduction. Yeah, because this we also tested in mouse. So if we deplete CD8 T cells, we don't get a response with anti-clever blockage. Yeah.
Thanks.
Yeah, you're welcome.
Perhaps I could also ask, if you don't mind.
No, of course, please do.
Hey Maija, nice to see you, and great talk. Great
Thanks.
to see the whole history of the Clever One story. But now that you know a bit about what the Mac4Me project is supposed to do, so what is your recommendation, what we or the doctoral candidates should be doing to follow your expertise here?
That's a difficult one. Yes. I think I always emphasize going to models that are translational, have impact in translation. That's why my lab does very rarely any mouse work anymore. So we try to adapt our systems into human settings. So we always use like primary cells, no like overexpression models. I hate those. Like I really want to study things in their like exact environment. So therefore, like if you can achieve patient material, primary cells, HLA matched donors, so that there is no incompetence in there, that you get like a readout that is not true because it's related to MHC activation or problem. So be wise, I would say, and consider what your model is giving, what information your model is giving you, and use the right models to answer the right question. I think that this is something that I know it's vague, but still, yeah.
That's a very good recommendation. Thanks, Maija.
So can I follow up on that? So regarding aging, this is an issue and one of the topics of Mac4Me. So did you look in your mouse models maybe to the aging of the system in reflection of its response to your treatment?
So we did not in mouse, but the interesting thing is that when people age, the more they have soluble Clever-1. So maybe it's causing some kind of immunosuppression or related to chronic inflammation that then is releasing soluble Clever-1. But that's interesting. So the most important factor that was increasing soluble Clever-1 in the circulation was cancer, but the next thing was age. Yeah, so that's an interesting aspect. But I have to say that for our drug and Clever-1, mouse is not necessarily the best model organ or the animal, due to the fact that mouse monocytes are not very Clever positive, whereas human monocytes are very positive for Clever. So that component is missing.
Yeah. Okay. Well, we certainly need to improve the models that we want to do in Mac4Me. So wonderful.